Hello, my dear students. Welcome into the first lecture on conductometric titrations. In this video lecture, we are going to learn the principle of conductometric titrations and the conductivity cell. Okay. In the previous lecture, we have completed the chapter pH metry. In this video lecture, we are going to see principle of conductometric titrations and the conductivity cell. Okay. First of all, what is mean by the conductance? Okay, you might be know the conductance of a solution means the ability of a solution to conduct what? Electrical current. What is the definition? The ability of a solution to conduct electrical current, that is conductance of a solution. Another definition of that conductance is what? The electrical conductance of a solution, which is measure of the conducting capacities of all the ions in the solution, can be used as a means of determining the equivalence points of titrations. Such a method is called conductometric titrations. The process of determining the quantity of a sample by adding measured increments of a titrant until the end point is reached. Okay, this is about the conductometric titrations definition. Okay, now we will see its principle, actual principle. Okay, but before that, see, this is the picture of the conductimeter. Left hand side, the display unit is there in red color. 120 is saying, right, that will be the conductance of a solution, and right hand side in beaker they have kept the solution, in that inserted the conductivity cell. The conductivity cell we can discuss later. Orange color conductivity cell, that will be connected to the conductometer, and this is the assembly to measure conductance of a solution. Okay, now principle of conductometric titrations. During a titration process, one ion is replaced with another, and the difference in ionic conductivities of these ions directly impacts the overall electrolytic conductivity of the solution. The principle of conductometric titration is based on the fact that during the titration one of the ions is replaced by the other, and invariably these two ions differ in the ionic conductivity, with the result that conductivity of the solution varies during the course of titration. The equivalence point may be located graphically by plotting the change in conductance as a function of the volume of titrant added. Okay. The principles of conductometric titrations can be explained most simply with reference to a particular case. Suppose we will consider example the titration of a very dilute solution of HCl with a NaOH of solution. That means in this titration we are taking hydrochloric acid, diluted hydrochloric acid into the conical flask or beaker, and NaOH we are going to fill into the burette. This is our actually titration with same concentration. See, this is the reaction in between HCl and NaOH. Obviously, when acid reacts with base, it which gives what? Salt and water. That is H plus plus Cl minus plus Na plus OH minus, which is gives Na plus plus Cl minus plus H2O. This is the reaction happens in between this titration. Okay. The variation of conductance during the course of titration in an ideal case can be calculated from the ionic conductance. Okay, first of all we must know each of these ion having particular conductors, that must first of all we know. Okay, if the solution of HCl contains equivalent weight of HCl, the theoretical conductance of the solution, assuming it to be a infinity dilution, that means the theoretical conductance of the solution will be, that means in this uh, titration our titrant solution, that is HCl, which having the specific conductance of each ion, that means H plus having particular conductors, Cl minus having another different conductors. Okay, that we will see now. See, lambda H plus ions, that is the conductors of H plus ions, plus lambda naught Cl minus, which is gives 349.82 plus 76.34, which is gives 426.16 mho into centimeter square. This is the unit of conductance. That means H plus ion, which is having theoretical conductance, is what? 349.82, and the theoretical conductance of Cl minus ion, which is having 76.34. Okay, when we add this one, we will get 426.16 mho into centimeter square. This is the conductance of both the ions, addition of both the ions. Okay, now similarly we can see for Na plus and OH minus also. When the acid is titrated with NaOH, the conductance will go on changing. Why changing? Because the H plus and Cl minus ions goes on decreasing. That we will see later, goes on changing. And when the acid is just neutralized, that is at the theoretical point, the solution will contain only NaCl. The conductance of the solution will be now. See, lambda naught Na plus plus lambda naught Cl minus, that is conductance for Na plus ion and conductance for Cl minus ion, that is 50.11 plus 76.34, which is gives 126.45 mho into centimeter square. These are two conductors of both the solution with separate ions. When an excess of 1 gram equivalent weight of NaOH is added to the solution, it will now contain Na plus NaCl plus NaOH. Okay, that means when we add excess after the neutralization, that means after the equivalence point, when we add extra sodium hydroxide solution from the burette into the beaker or conical flask, at that time what will happen? In that solution, that means in titrant solution will contain what? NaCl plus sodium hydroxide. Then the conductance of the solution would be, see, in the beaker or conical flask, what will we get? NaCl plus NaOH. Okay, then the conductance for that, lambda naught Na plus plus lambda naught Cl minus plus lambda naught Na plus lambda naught OH minus, which is gives 50.11 plus 76.34 plus 50.11 plus 198, which is gives 374.56 mho into centimeter square. So entire at the after the equivalence point, we will get the conductance 374.56. Okay, if this conductance is plotted against the volume of NaOH added, a titration curve like that of ABC in figure will be obtained. See the figure, how the figure will obtain when we plot the conductance versus volume of NaOH solution added graph. Okay, see like this we will get the graph. Okay, see, just we have seen the procedure. Now Now I will again explain the procedure. Suppose at the initial we are taking diluted hydrochloric acid solution into the beaker. Suppose we are taking 10 ml. In that solution, the H plus ions and Cl minus ions is there. When we add the sodium hydroxide solution from the burette, at that time the some amount of HCl solution will be used to convert NaCl plus H2O. Okay, but at the initial stage the conductance will be very high at the A position. Okay, why high? Because of the ions of H plus Cl minus will be high in quantity. Okay, that's why the mobility of the ions will be higher. The conductance or the electrical current in the solution also higher. That's why the conductance also higher. When we add the NaOH solution from the burette into the HCl solution, the H plus ion and Cl minus ion goes on decreasing. Ultimately, what the conductance of solution also goes on decreasing. Okay, up to the B point. Here B point we will get the end point of titration, but when we add NaOH solution from the burette excess, what will happen? Again Na plus OH minus ions also increases. Okay, and in the solution contains NaCl, that is salt. Okay, when we add excess amount of NaOH from the burette, again the conductance goes on increasing up to the C. Why? Because of Na plus OH minus also conducting the electrical current. Obviously conductance of a solution goes on increasing. That means we will get the V-shaped graph at the end. So this is the principle of conductometric titration. This is the graph of that conductometric titration, V-shaped graph. Now we will see the conductivity cell, but before that the descending portion AB representing the conductance of unneutralized acid and salt, while the ascending portion BC representing the conductance of salt and excess alkali added. The point of intersection of the two straight lines will be the equivalence point of the titration. In an actual titration, the lines become slightly curved, particularly near the equivalence point, mainly due to increase in volume of the solution and intrionic effects. This is all the more so if the salt formed undergoes hydrolysis or the product form is appreciably soluble in precipitation titrations. Just before this slide, I tell entire procedure. Okay, so this is the remaining part of that graph explanation. Okay, now we will see conductivity cells. Okay, so this is the figure of conductivity cell. In the left hand side they in blue color they draw the diagram. In that diagram the square shape that will be platinized platinum electrode. Okay, that both the wire connected to the conductimeter. Okay, just before to slide we have seen the diagram or the picture of the conductimeter. In that the orange color conductivity cell we have seen that conductivity cell we are going to connect the conductivity meter. Okay, and left hand side how the platinum wires are seeing actually in solution that they have shown. Both the wire will be coated by platinum, that means platinized platinum electrode both, and that will be dipped into this solution which is having we are measuring the conductance. Okay, a conductor cell is a vessel of high density plastic material fitted with two uniform platinum coils fixed parallel to each other. That means both the plates, platinized platinum plates are kept in a fixed parallel line with each other. A conductance cell which is convenient for conductometric titrations is shown in above figure. The cell must be handled with care so as not to alter the distance between the two electrodes. For a given cell, the distance L between the electrode and the area A of the electrode are constant. For a given cell, ratio of L by A is a constant and called the cell constant. In solutions of low conductance, the area A of the electrode should be larger and the distance should be small, that is to say the cell constant must be small. For solutions of high conductance, the area should be small and the distance between the electrodes should be more, that is to say platinized platinum electrode, the cell constant should be high. In order to electrodes eliminate as far as possible the polarization effects due to the alternating current, the electrodes are electrode deposited with a platinum black. Such electrodes are called platinized platinum electrode. Finely divided platinum coating provides a large surface area and thus catalyzes the combination of hydrogen and oxygen which tend to get liberated when an alternating current is used. The polarization EMF is thus practically eliminated. So thank you dear students. We have seen the principle of conductometric titrations and conductivity cell. In the next video lecture, we are going to learn the different types of conductometry titrations, its applications and limitations. Okay, thank you.